Hello guys, in this video, genetics part 2 will be my last video for genetics. Due to that genetics details are so specific and are mostly the same, I'll be only doing genes, assortment of genes, and traits in this video. Independent assortment. Mendel crossbred yellow peas with wrinkled green peas, and what he got was a round yellow peas. This shows that allele for round yellow peas was dominant over the green ones. This cross of plants created hybrids needed for the next cross. However, this cross does not indicate whether genes assort or segregate independently. Mendel knew that the F1 plants were heterozygous for both the seed shape and seed color genes. Independent assortment continued. The principle of independent assortment states that genes for different traits can segregate independently during the formation of gametes. Independent assortment helps account for the many genetic variations observed in plants, animals, and other organisms. Summary of Mendel's principles. His principles solidify what we have today. The inheritance of biological characteristics is determined by genes. They are passed on from their parents in cases there may be a dominant recessive version. In each sexually producing organism, each adult has 2 copies of each gene, one from each parent. Dominance. 2 4 o'clock plants, a red and a white, will breed 4 pink flowers due to incomplete dominance. In this case, a heterozygous is somewhere in between the 2 homozygous phenotypes. A similar situation is codominance, which is when both alleles contribute to the phenotype. With each combination, it creates new designs that carry both phenotypes. Polygenic traits. They are traits controlled by having many genes. Different combinations of these genes includes a variety of traits. For example, the wide range of human skin color is partly because more than 4 different genes control this one trait. Genetics and the environment. The characteristics of any organism are not determined by the genes it inherits. Rather, it is determined by the interaction of genes and the environment. For example, genes affect a sunflower's height and the color of its flowers. However, these same characteristics are also influenced by climate, soil conditions, and the availability of water. Genes provide a plan for development, but how that plan unfolds depends on the environment. Griffith's experiments. Griffith is a British scientist who was trying to figure out how bacteria make people sick. He injected lab mice with disease-causing strain, and they died of pneumonia. He then made a heat-kill version, and he found out that they don't produce a chemical poison. He had another experiment where he injected a heat-killed and disease-causing. This time they died because the disease-causing bacteria have put their genes into the heat-kill bacteria. Griffith's conclusion. Griffith concluded that the heat-kill bacteria and the disease-causing bacteria, some factor transfers from the heat-killed into the live cells. That factor could change a harmless bacteria into new disease-causing bacteria. Since the ability to cause disease was inherited by the transformed bacteria's offspring, the transforming factor might be a gene. DNA. We humans have been using selective breeding to make the traits in animals what we want for thousands of years. When we discovered DNA, we used the genes to make our own traits in animals, like more muscle in cows and bigger chickens. Nearly all crops and livestock have changed drastically since they were domesticated. You guys can go read more about the details of DNA in an article or some text, because most of the information is usually the same. Thanks for watching, guys. Even though this genetics video series was supposed to be long, the plan turned out that most of the information was actually very similar to each other. My next video will be about evolution, and I'll be explaining Darwin's theory and all the other scientists who came up with new theories.